Welcome, everyone. Welcome to the OTS webinar series. So today is a special moment. So as I think most of you know, the OTS journal is Nucleic Acids Therapeutics. And recently it was decided that there will be publications highlighted in the OTS webinar series. And it are the journal editors. So that's Bruce Sullinger, Annemiek Aatsma-Rus, and also Graham Parker, who selected this particular paper to be highlighted in both the webinar series, but it's also highlighted and it will be open access on the NAT website. So it's my honor to introduce Sarah Ellen. So Sarah Ellen is a PhD candidate in the Corova lab in UMass, and she will highlight her publication on mRNA nuclear clustering that leads to a difference in mutant Huntingtin mRNA and protein silencing by siRNAs in vivo. So Sarah, the floor is yours.
Thank you. Good morning, everyone, and thank you for having me. As stated, my name is Sarah, and I'm a PhD candidate in the lab of Dr. Anastasia Korova at the RNA Therapeutics Institute at UMass Chan Medical School. And today I'm going to be discussing some of our recently published work regarding the impact of mRNA nuclear clustering on siRNA-based silencing efficacy for both mRNA and protein. So just to begin with, the sort of background for this project is working to develop siRNAs as therapeutics for Huntington's disease. Huntington's disease is an autosomal dominant inherited neurodegenerative disease and is generally marked by a loss of motor control in adult onset, both cognitive and emotional decline, a death of the medium spiny neurons within the striatum, and this aggregated Huntington protein, as well as clustered Huntingtin mRNA. So this is caused by a mutation within exon 1 of the Huntington gene. Particularly, it is caused by the accumulation of an increased number of CAG repeats within exon 1. And this increase in CAG repeat number is actually correlated with an earlier age of onset. So if you have more CAG repeats, generally you have an earlier age of disease onset compared to having a fewer CAG repeats. So just to sort of go over the molecular basis of disease a little further, generally people with Huntington's disease have inherit one wild type allele of the Huntington gene, so with fewer than 40 CAG repeats, and one mutant allele of the Huntington gene, greater than 40 CAG repeats. The gene undergoes somatic expansion over time, and this causes an accumulation of CAG repeats up to 60 repeats and then 150 repeats. It gets greater over time. And this somatic expansion of the DNA is then translated into the mRNA of Huntington. So the mutant Huntingtin mRNA is translated, but not only this, but this expansion results in an alternative splicing event that creates an alternative transcript. This transcript consists of exon 1, where that expanded CAG repeat is, as well as a bit of intron 1 around 7 kb. And this is a transcript we term HTT1A. Both of these transcripts accumulate within clusters in the nucleus. The HTT1A specifically is found predominantly within these nuclear clusters. And then both transcripts are translated into protein. This is interesting for the HTT1A transcript, as this transcript is polyadenylated and exported from the nucleus in a way that one might not expect from this alternative transcript. But both transcripts form proteins and then form protein aggregates. And generally, it's thought that a combination of these different proteins and these mRNA clusters leads to neurodegeneration down the line, though the exact mechanisms which govern the toxicity of Huntington are not exactly known or well characterized quite yet. So the working model for how Huntington's disease develops currently is that this somatic expansion facilitates the generation of toxic Huntington species. So over time, you have the accumulation of more and more CAG repeats, and recent findings from the McCarroll lab demonstrate that we actually don't see toxicity until you've accumulated 150 plus repeats. So a great number from what you would have initially had. And there appears to be some sort of threshold where this toxicity really takes place, as well as a threshold in which somatic expansion, the rate of somatic expansion increases greatly. So in order to study Huntington's disease, we have a few different mouse models. There's no one particular mouse model that recapitulates every single phenotype of the disease, and therefore there's no gold standard Huntington mouse model. One of the mouse models that's most widely used is these knock-in models termed the Q series. This Q allelic series contains an insertion into exon 1 of the endogenous mouse Huntington gene, as well as a bit of the intron inserted. And the number of repeats in the Q series varies. So there's Q20 mice with 20 CIG repeats all the way up to Q175 mice. They can be heterozygous or homozygous for this insertion. However, they do not experience any phenotypes of Huntington's disease until greater than 1 year of age, with the exception of the Q175 that have a bit earlier of an onset. They also do not possess human intron 1 except for the first 200 bases, and therefore they don't really get the severe sort of clustering phenotypes that we've been interested in studying in Huntington's disease. So we've sort of opted for looking at instead these transgenic models of Huntington's disease. One of these is the YAC128 model. So this contains the full-length human Huntington transgene. The gene has 128 mixed CAA and CIG repeats. However, at its longest stretch, it only has around 81 uninterrupted CIG repeats. Therefore, this model doesn't really undergo significant amounts of somatic expansion, which is sort of a key feature of disease that we'd like to be able to replicate in these models. However, the nice thing about these models is because they contain around 4 copies of the human transgene, we get a much earlier age of onset, so between 6 and 9 months for disease phenotypes to set in. We also have a fully humanized intron 1, so we see really profound amounts of clustering due to both the high expression of the gene and the humanized intron. The other model that we'll be looking at today is the BAC-CAG model. This model also contains full-length human Huntington inserted as a transgene, and this model actually has over 120 uninterrupted pure CAG repeats. However, the expression to this model is rather low comparatively. It only has 2 copies, and they're not incredibly highly expressed. So the age of onset for phenotypes is a bit later at about 1 year old. And then they do also have humanized intron 1, but because of the low expression, we get very low levels of that HTT-1A alternative transcript. So it can be a bit tricky to measure at times. However, they do see some clustering, which is useful for the research we're discussing today. And yeah, again, I'd like to focus on these particular transgenic models as they are sort of the subject of the paper we published. Now, the nice thing about these transgenic models also is that because they contain both the mutant human Huntington with that expanded CAG tract and the endogenous mouse Huntington with that much shorter CAG, because of the species difference, you can measure the mutant and the wild type Huntington separately. So we can design specific probes to individually measure both the mutant and the wild type Huntington, so we can see whether or not they're behaving differently in different contexts. Human Huntington, when looking at it in relation to the wild type Huntington, we see that this wild type mouse Huntington is dispersed fairly evenly between the cytoplasm and the nucleus. We also see that it seems to accumulate in individual foci, or these little red dots here. And let's see, just get a laser pointer here. But when we look at the human Huntington in green throughout this presentation, you can see that it is slightly more shifted toward the nucleus, with a slightly greater nuclear localization compared to the mouse Huntington. And you can also see that it accumulates in these sort of characteristic clusters within the nucleus. And particularly when you compare it to the mouse Huntington, you can see that these clusters are present in the human mutant context, but not in the mouse context. Additionally, the nuclear fraction of human Huntington is higher than mouse wild-type Huntington, and this is all shown from images within YAC128 mouse brains. So just to show the quantifications for this between a number of tissues, we see that there is an increase in the nuclear localization of mRNA foci when looking at the human Huntington specifically, and this goes up with age a bit as well. And then we see that there's an increase in the general volume of the clusters. So you can see that there's clustering happening in the human Huntington that's not happening in the mouse Huntington. So the other sort of characteristic of disease that we look at is this human HTT-1A. The human HTT-1A, so this is exon 1 and intron 1, it also forms clusters that localize to the nucleus, and it is very nuclear as compared to the human Huntington that just has a nuclear bias. The HTT-1A fragment is highly nuclear and highly clustered. And you can see, interestingly, that these clusters seem to co-localize with the full-length mRNA. So when you look at individual foci for this HTT-1A fragment and for human Huntington, you see some overlap, but they're not really co-localized readily. Whereas the clusters, so this collection of individual transcripts clustered together, we see that it's highly co-localized with the full human Huntington. So this is suggesting that both of these transcripts together form a singular cluster. And one of the hypotheses in the field is that the HTT-1A fragments are working to seed the formation of these full-length clusters. Another feature of the disease is that we see these full-length expanded Huntington protein aggregates, which also form within the nucleus of cells. And these have been fairly well characterized for quite some time now as a feature of Huntington's disease. So what we use in the lab is we want to look at the impact of, in order to better elucidate the impact of Huntington, we want to look at being able to disrupt it with siRNAs. So siRNAs function via the RNA interference pathway. They associate with AGO2 to form the RISC complex. The RISC complex then localizes to the strand of interest and then cleaves it, silencing the gene at the mRNA level specifically. We also use a number of different chemical modifications in order to stabilize the molecule, adding greater potency, efficacy, stability. So for CNS applications, we use these divalent siRNAs, which were developed in the lab and are two siRNA linked by this hydrophobic linker here. We also use a number of chemical modifications. They're fully chemically modified siRNA, and we use 5' modifications such as vinyl phosphonate, 2' ribose modifications such as the fluoro and O-methyl RNAs, phosphorothioate backbone. And then we also use these 3' XNA modifications as developed by Ken Yamada's group in the lab. So we have also published some work, this Altman et al. Paper, in which we've characterized a divalent siRNA specifically against Huntington. And you can see that at up to 6 months, we are detecting, up to 6 months post-injection into the ventricle, we detect significant amounts of divalent siRNA accumulated in the brain. And then we can also see that we get a significant knockdown of mRNA and protein, definitely at 1 month, in most regions still at 4 months, and then in some regions up to 6 months post-injection. So we're getting really significant silencing of Huntington with our compound. The other nice thing about this divalent siRNA is that it is cross-homologous to the human and mouse sequence for Huntington. So we have full cross-homology here. So we expect to have relatively similar silencing of both transcripts. And this is very nice for these mouse models that we can look at both the humanized Huntington and the mouse Huntington. So to summarize, we have a number of accessible animal models where the wild type and mutant Huntington are expressed. We also have assays to look at both mutant and wild type Huntington separately due to the differences in sequence there. So we have protein assays that we measure via the automated simple Western. We are able to measure bulk mRNA using this quantagene branched DNA assay, which uses micro wells and capture probes in order to bind the specific mRNAs to a well. And they are then amplified with a series of labeling steps, and then we can read the bulk fluorescence of the mRNA from there. And we have probes specifically for the mouse Huntington and the human mutant Huntington. So we can look at those both separately. We also have an assay to measure the intracellular localization of the mRNA. So here, this RNAscope fish-based assay from ACD Bio, we can see the human Huntington in green and then the mouse Huntington in red. So we can use tissue to then similarly do a series of amplification steps and be able to visualize both with a different fluorescent tag while in brain sections on slides. And as shown previously, we've developed an siRNA that targets both mutant and wild-type Huntington at the same location. So using these tools, we are able to look at the impact of Huntington lowering on the protein, mRNA, and mRNA localization of both wild-type mouse Huntington and mutant human Huntington within these models. So the initial sort of construct that of data we have is in the back CAG mice. So just to remind everyone, these are the mice with over 120 uninterrupted CAG repeats. They have the human mutant transgene, and they also have their wild-type mouse Huntington. They were injected at 16 weeks old, and then they were euthanized at 20 weeks. So if we look at the mRNA knockdown following injection, we see that wild-type Huntington in the striatum is significantly silenced around 60 to 70% here. In the cortex, we see very similar levels, silencing around 70% at the mRNA level. But when we look at the mutant human Huntington, we see no significant silencing in the striatum or the cortex. So this is kind of an interesting difference given that we have cross-homology between the compounds and they should target both transcripts relatively equally. When we look at the protein data, we actually see that there's very similar and potent knockdown of both the wild type and mutant Huntingtin within the striatum. And we see this in the cortex as well. So this is true across regions. We also, so I suppose when you look at it all together, we see a really great difference between the degree of silencing from mRNA to protein. So the mRNA wild type, the wild type mRNA is being silenced around 75%. The mutant mRNA is not being significantly silenced at all. But when you look at the protein silencing, we see around 80 to 90% silencing of both wild type and mutant Huntingtin. So this is a really interesting difference that we start to see. So this is a difference we see in one model of disease, but we want to know whether or not it was applicable just generally. Is it applicable to Huntington in a different model context? So looking at the YAC128 mice, these are the mice with around 81 uninterrupted CAG repeats. We injected them at 4 weeks, and then they were euthanized at 28 weeks, much later. So this is a longer time point. And at this time point, we saw around 50% reduction of wild type Huntingtin within the striatum. When we look at the cortex, we see very similar knockdown, around 50% lowering of wild type Huntingtin. But once again, when we look at the mutant Huntingtin, we see around 25% silencing at most. It does reach significance this time in the striatum, but it is not significant in the cortex, and it is much less silencing than we see compared to the wild type Huntingtin mRNA. So once again, we are able to look at the protein, and we saw significant reduction of both the wild type Huntingtin and the mutant Huntingtin within the striatum and the cortex, even at this longer 6-month time point. And once again, when you compare the degree generally of mRNA knockdown to the protein knockdown, we see much greater silencing of the protein with these siRNAs. So now we see this in 2 different models of Huntington's disease. For Huntington, we see this difference in mRNA versus protein silencing, and we also see a difference between the mutant and the wild type mRNA silencing. So again, to summarize what we've found so far, the lab has a compound which targets both mutant and wild type Huntingtin. There is a difference in the degree of mRNA silencing of the mutant and the wild type Huntingtin. However, there is a potent silencing of both mutant and wild type protein. And we want to know why this is the case. So generally, we know that only cytoplasmic mRNA is translated into protein. So the hypothesis here was that we also know CAG expansion alters the localization of Huntington to the nucleus. I showed this data earlier, but again, we can see here that the human Huntington mRNA in green has much greater nuclear localization than the mouse Huntington. It's also forming these clustered structures within the nucleus. So we're able to measure this with RNAscope. This then measures the transcripts separately, staining the human mutant Huntington in green and the wild type mouse Huntington in red, so we can look at where these transcripts are in the cell. So this is a testable hypothesis now, and we propose that CAG expansion changes the fraction of mRNA in the cytoplasm and thus changes the accessibility of that mRNA to siRNA-based silencing. So here's the data from testing that using the fish-based assay. We have the nuclei of the cells stained with DAPI in blue here. And then once again, we have mutant human Huntington in green and wild type mouse Huntington in red. So when you look at the mouse Huntington of the NTC treated brains, we see again fairly dispersed staining of individual RNA foci. So you don't really see much clustering, and you see a relatively even distribution between the nucleus and the cytoplasm. When we look at the human mutant Huntingtin, we see these really strong clusters within the nucleus, and we see a slightly greater distribution of transcripts in the nucleus compared to the cytoplasm. So when we look at brains treated with our Huntington siRNA, we see just generally fewer transcripts, which is good, as it confirms that our treatment is working. And when you look at specifically the wild type mouse Huntington, we see much less foci staining here. You see that most of the red transcripts here are wiped out, especially in the cytoplasm. But when you look at the mutant human Huntington in green, we see that there is some resistance to the silencing, and that particularly the clusters structured here are still left over within the nuclei of cells, as are many of the nuclear transcripts. So again, we looked at the same back CAG mouse tissue, so 120 CAG repeats, and these images were all taken in the striatum of the mouse brain, as this is the area of interest and area most impacted by Huntington's disease. The mice were taken down 4 weeks post-injection into the ventricle. And the quantifications for the images shown previously are here, where we can see that the wild type mouse Huntington was significantly lowered overall. When we look at specifically the cytoplasmic transcripts, we see really solid and significant lowering. However, when we look at nuclear transcripts, we also see significant lowering, which is great. But it's slightly lesser than the cytoplasmic silencing, but we still see really significant reduction of wild-type mouse transcripts in the nucleus. We also see significant silencing of the mutant human Huntingtin mRNA, though to a lesser degree overall than the mouse mRNA. We see a significant reduction of the cytoplasmic transcripts, but we see no significant silencing of the nuclear human Huntingtin mRNA. There's some lowering, but it does not reach significance. So we confirm that the CAG expansion has changed the fraction of mRNA that is available to the to siRNA-based silencing through both nuclear localization and clustering. And though we showed this with one target in 2 different mouse models, we do know that incomplete mRNA silencing resulting in really strong protein lowering in vivo seems to be a widespread phenomenon. So it seems to be applicable to more than just Huntington. We have another project for Huntington's disease in the lab looking at targeting different mismatch repair factors to try to sort of slow or stop somatic expansion before it begins. So this is targeting all of these factors. And another student in the lab, Jillian Belgrad, has done a lot of work to develop siRNAs for each and every one of these transcripts. So just showing some of her data quickly, she screened a number of targets, and you can see that she gets around 50% silencing of the mRNA at most of these transcripts. However, for all of the transcripts shown, we get much greater protein silencing than mRNA. So we see lower than 25% silencing for many of the targets, and for all of these targets, we get greater than 50% silencing. So it is not only a phenomenon that impacts Huntington specifically, but a number of transcripts seem to have much greater protein silencing than mRNA silencing. This seems to be a widespread phenomenon that impacts siRNA-based silencing measures, and we believe that it should be taken into account generally that if your mRNA silencing isn't that great, maybe it looks much better at the protein level. But sort of our conclusion here is that it is important to look at both mRNA and protein as an outcome when using siRNA because this discrepancy may be present. We also believe this discrepancy is more pronounced in complex tissue like brain or muscle. So all of these studies have been done in brain tissue, and in brain and muscle, nuclear retention of mRNA is more common. And sort of the final consideration we'd like to make of this is that other repeat expansion disorders do have strong impacts on the CNS and may require similar consideration in regard to the development of gene therapy-based therapeutics. So Huntington's disease is not the only CAG repeat expansion disorder. It's one of many. So SCA and DRPLA are both other disorders that impact the central nervous system and are caused by CAG expansions. And CAG expansion disorders are not the only disorders that are caused by somatic repeat expansion. We have other disorders such as C9orf72, myotonic dystrophy type 1, and Friedreich's ataxia that are all caused by repeat expansion of a specific repeat and cause neurological disease. So this may be a consideration for a number of these diseases as they're all marked by a very similar nuclear clustering, and the underlying biology has a lot of similarities. So when developing therapeutics, we just believe that the localization and structure of the target should be taken into consideration, and might just be an additional challenge when it comes to developing siRNAs against these targets. With that, I'd like to thank everyone that's been working on this project, particularly our sort of Huntington's disease research group. So my PI and mentor, Dr. Anastasia Kurova, as well as Jillian Belgrad, who did all of the mismatch repair work that I showed a bit of. Our animal team, Eric Liu and Ashley Summers, and then the Kurova Lab alum that set the basis for this project, Dan O'Reilly and Soki Lee. I'd also like to thank the DeFiglia Lab for all of their wonderful protein work that they were able to do for us. It's very helpful. And then the Aronin Laboratory, Rachel and Joe did a lot of this work on the YAC128 mice as well. So thank you. And I believe we have plenty of time for questions.
Yes, Sarah, thank you for this excellent presentation. I really enjoyed it. So if you have a question for Sarah, please type it in the Q&A. And I think that I can start with a question. So obviously, next to the siRNAs, you have other kinds of RNA therapeutics. So for example, gapmers. And to my knowledge, gapmers do enter the nucleus and can break down the RNA in the nucleus. So would using a gapmer approach, would that help you by understanding the mechanism?
Mm-hmm. So there has been some other research in the lab showing that, well, obviously ASOs are able to enter the nucleus, which is a great advantage for these nuclear transcripts. They don't do anything to impact that clustering. So the clusters are still resistant to ASO-based silencing, which is a problem. But the nuclear aspect of it can be somewhat mitigated by using an ASO like a gapmer instead. So that is an approach we're looking into as well, trying to look at ASOs in addition as a different silencing methodology.
So when I heard this entire story, I would say, so do you still think that Huntington's disease is a protein gain of function disease? Or do you think, well, for example, you have the Friedreich syndrome, where it's more like an Friedreich ataxia syndrome, where you have like an RNA gain of function model, where the RNA kind of folds and attracts all kind of proteins so that cannot perform their normal function. Do you think this might be a mechanism for Huntington's disease as well?
So yeah, it was definitely traditionally thought of a protein gain-of-function disease, but I think the field is really learning that it is so much more complicated than that. It's not as simple as just a mutation that leads to a toxic protein. There was a study that showed that actually it is not the number of glutamines, so the polyQs that contributes to earlier disease, but rather the number of CAGs itself. So if you have CAAs, it doesn't lead to an earlier age of onset the way uninterrupted CAGs do. So while we don't really know the impact of the mRNA itself, so we don't really know whether or not these clusters are pathogenic or whether they're protective. These are still outstanding questions, but we do know that the mRNA does have some role, and that this is not, I think we need to start treating these repeat expansion diseases as their own class of disease, where a lot of the toxicity is caused by that expansion. So that's why a lot of the current therapies being pursued are more like what I showed just at the end there, where they're targeting the mismatch repair pathway to try to reduce the amount of expansion before it ever happens. So by acting at that earlier level, I think that's kind of more of the causative agent of disease, if that makes sense.
Yeah, I think to come back to this, so I think so when we think about the somatic repeat expansions, this is something that occurs on a DNA level, right? So this will continue anyway. So do you think that the RNA targeting therapies, do you think that that makes sense? Because I think in one of your introduction slides, you show that it only starts after 180 repeats or even higher. So is stopping the somatic expansion is not more important than lowering the RNA or protein levels?
Yeah, it can certainly be helpful. I think the idea of stopping somatic expansion. However, it does seem like, given some recent studies, that there isn't really toxicity of the mRNA until after 150 repeats. So if we're able to, one option is lowering that somatic expansion, since you're only inheriting a CAG repeat of around 40 or so, 40 plus. That it's not toxic yet. And if you act at that point, you can stop it from ever expanding. But I think we're looking at additional sort of downstream therapeutics to try to figure out what exactly is the cause of toxicity. Is it that HTT transcript? Is it the clustering? And can you act a little later on also? So I think there's a lot to be learned about the biology of the disease that'll allow us to better develop therapeutics.
So I'm moving to the questions in the chats. So if you have a question, please use the Q&A. Yeah, so first question, I think you already answered that question. So have you tried ASOs?
For full length, we're working on developing ASOs. There's also, I believe, some ASOs that have moved to clinic for Huntington silencing, and we're interested to see how they do from there. But
yeah, maybe to
it's a thing that's being worked on.
Maybe expand to this question. So I believe there are quite some ongoing clinical trials for Huntington's disease. So how does your approach fit into the entire field?
Well, so we are an academic lab, so a lot of our approach is just developing tools to greater understand the biology of things. And we hope that those our findings about the biology of the disease can help to inform the development of therapies by other things. But I think it's really important because we don't know the impact of clustering and we don't know the impact of HTT to start to learn those things so that we can have a fully informed understanding of the disease before we really before anyone's going into patients.
So about this HTT So how much is known about that? Is it translated into a protein? Is it translated into different reading frames, like the run translation you see in other repeat expansion disorders? Is it also, is it clustering? Does it have a different, different transcripts? So any idea about that?
So yes, it's translated into a protein. And that protein actually is very, very similar to the protein fragment present in the R62 mice. This is a mouse model of Huntington's disease that is highly pathogenic. So this mouse model has really severe phenotypes and dies a lot faster than other models. So that's sort of one of the driving hypotheses to why it's so toxic, or to why it may be a toxic factor, is because it's very similar to that. We do know it clusters as well. That's another thing we know. But I do think much research remains to be done. A lot of the research on this HTT1A fragment has been done by Joe Bates' lab in the UK. And they've been pioneering really the discovery and importance of this fragment. So I think much remains to be seen, but slowly they are coming out with more and more evidence that this might be a compelling feature of disease and not some strange byproduct. But
thanks. So another question from Hans, and I think we already discussed this a bit. Does nuclear clustering of the mutant HTT mRNA contribute to the disease phenotype? For example, by sequestration of splicing factors or silencing of cytoplasmic mutant mRNA, sufficient for disease treatment. So, any ideas about that?
yeah, so we don't 100% know the answer. We don't know whether or not it is contributing to phenotype directly or whether it's protective. It could actually be sequestering this mutant mRNA in a way that is good because it's not being translated into the protein if it's being sequestered in the nucleus. But we do know that there's a lot of RNA binding factors that may bind to CAGs and that are impacted by this mutant Huntington. So that might be something that is contributing to pathology, but we don't know the answer. These are just a couple different hypotheses as to what could be happening here. But I think that's something that needs to be looked at.
Is there any data on? Did people already isolate these clusters and checked what's sequestered?
Yeah, I believe there is data, and I believe there's some association with like MLB and MBNL1 and other RNA binding factors. But yeah, there's certainly data that they associate with a number of different factors, but whether or not that is sufficient to cause disease, I think, is the question.
Thanks. Question from Damon. Great talk, Sarah. One question on our RNA scope probes detecting HTT1A. There might be two forms of HTT intron 1 in the nucleus. One form as intron 1 within HTT full-length pre-mRNA before splicing. The other form as intron 1 after it's spliced out of the pre-mRNA. Could you comment how abundant these two forms might be?
I hope I understood that question, but I believe you're asking how we control for the pre-mRNA being present, and that's certainly been a consideration. So one, the pre-mRNA should not be present outside the nucleus, and we definitely do see some HTT1A transcripts outside of there. And two, we've actually looked. So the intron 1 probes are all designed specifically for intron 1, and we've looked at if we design HTT1A only has the first 7 kb of intron 1. So we've looked at if we have a probe after that point, so after the polyadenylation site, where that transcript is no longer HTT1A, how abundant that is. And we do see that it is far less expressed than the HTT1A itself. So we've certainly done that control, but it can certainly be a tricky thing to measure to some extent. But we have looked at it, and it is independent of just being the pre-mRNA. But yeah, hopefully I addressed all of that.
Yeah, I think so. So just coming back, so What is known? So I think you mostly use the two different animal models. So what is known about, for example, post-mortem brain tissue of patients?
So there is some work regarding the postmortem brain tissue. There's a bit of work trying to identify those HTT-1A clusters. I believe Soki in the lab did some work on human brain tissue before me. And the Bates lab also has characterized the presence of that fragment in juvenile HD brains. But it is very hard to measure, particularly in human brain tissue, just due to the nature of it when it's not the most abundantly expressed transcript, and it can be certainly tricky to measure in tissue that's decayed a bit. But I think we're working on developing better and better antibodies to be able to really examine its presence in human brain tissue. So there's been some work on that, but it's definitely still a progressing field.
I'm asking because what I always think is, so when you get these somatic hyper expansions, these cells die, right? So you lose these kind of cells. So this might be quite difficult to show something that might not be there. That I think that might be quite challenging to show. So.
Oh yeah, certainly. Yeah, that does seem to be the problem. That the hypothesis generally is that these clusters and these fragments are very toxic, and therefore a lot of the cells that have them may or may not be present by the time you get postmortem brain tissue. The cells might already have died. There has been an attempt from the McCarroll lab, I believe, to characterize the presence of protein aggregates in cells and correlate them to the amount of repeat expansion. And I believe they have some data showing that they've presented at conferences showing that these clusters are present in much more expanded cells, so cells that are further along the line of disease progression.
Yeah, to continue with the protein aggregates. So I think that these animal models also have protein aggregates. And you see, almost 100% reduction of the Huntington protein. Do you also see a reduction of the protein aggregates?
So I haven't actually directly looked at that, although I know we have collaborations with some preliminary data showing that that HTT-1A lowering may contribute to the formation and the lowering of those protein aggregates. But I believe that publication is in progress. But I haven't directly looked at the protein aggregates with the full-length Huntington knockdown.
Oh, thanks. Question from James. Nice work. How do you explain the reduction in nuclear mRNA when the main site of action of the siRNAs is thought to be the cytoplasm?
So yeah, it definitely does. I thought that was one of the more interesting things that we found, that it seems like there is some silencing occurring in the nucleus. And it's hard to tell exactly why this could be happening. Some of it could be that there is, in fact, some activity of the RISC complex in the nucleus, even if it is extremely limited. It also could just be that there is a distributive shift of the mRNA localization, and that maybe just by disrupting a lot of the cytoplasmic transcripts, some of them are being exported from the nucleus as well, and it might just be just sort of an equilibrium thing that's happening. We don't really have a proven or direct explanation for either case, but it could be either, and I think that's one of the more interesting follow-up questions that someone might be interested in pursuing.
Thanks. Those were all the questions. Thank you all for joining. Sarah, thank you very much for presenting this very nice work. I think we see you in Montreal in a few weeks from now. Yep. So if you have any remaining questions for Sarah, of course you can send an email, but you can also talk to her at the OTS conference in Montreal in a few weeks. So thank you very much, and see you in Montreal.
Thank you.